In this final lesson on the nervous tissue, we will conclude with synaptic activity. We will look at the effects of neurotransmitters on their targets and conclude with the discussion on the capacity for different parts of the nervous system to experience regeneration and repair. Okay, let's look at what happens once the signal reaches the axon terminals and we have activity at the synapse. There are two types of synapses in the excitable tissue of your body. Electrical synapses involve the flow of ion currents from cell to cell through connective passageways called gap junctions. So this allows for faster as well as two-way transmission and leads to the synchronizing of neural or muscular activity. This occurs in cardiac cells as well as smooth muscle cells. Skeletal muscles and neurons experience excitability in a different way in that these have chemical synapses. Transmission of information is one way from the presynaptic neuron to the postsynaptic neuron, which might be another neuron or a skeletal muscle cell. And the chemical, which is a neurotransmitter, crosses a gap, which is the synaptic cleft, the space between the axon terminal of the presynaptic neuron and the membrane of the postsynaptic neuron. Most synapses in the body are chemical neurons. This diagram is great at depicting the events that occur at a chemical synapse. So once the action potential travels the length of the axon and reaches the axon terminals, that depolarization wave will cause the opening of localized calcium channels in the synaptic end bulbs. Calcium concentration is higher outside the cell, so calcium will flow into the cell down its concentration gradient through voltage-gated calcium channels. These channels only open when the voltage changes due to the arrival of an action potential. The influx of calcium then stimulates synaptic vesicles inside the synaptic end bulbs to move towards the membrane, fuse with the membrane, and release neurotransmitter by exocytosis. The neurotransmitter will diffuse and travel across the synaptic cleft and bind to receptors on the postsynaptic neuron membrane. So those receptors would be associated with ligand-gated channels that open in response to the binding of the ligand, and depending on which ion is now allowed to flow into or out of the cell due to binding of that neurotransmitter ligand, you may get an excitatory postsynaptic potential, which is more likely to cause the postsynaptic cell to experience an action potential, or, again, depending on what happened, it may cause an inhibitory postsynaptic potential where the membrane is actually hyperpolarized, moves away from threshold, and essentially the activity in that postsynaptic cell is suppressed. This is all very similar to what we saw at the neuromuscular junction. That is exactly what's happening at the neuromuscular junction, where a nerve impulse along a somatic motor neuron causes an influx of calcium that causes synaptic vesicles to move to the membrane and release the neurotransmitter, in that case acetylcholine. Acetylcholine binds to receptors on the postsynaptic neuron, which would be the sarcolemma if we're talking about the neuromuscular junction, and that opens up sodium channels, leading to the influx of sodium, which now creates a postsynaptic potential. So here we have it described for us where the action potential reaches the end bulbs and opens voltage-gated calcium channels. The inward flow of calcium triggers the release of neurotransmitter from the synaptic vesicles. The neurotransmitter crosses the cleft and binds to ligand-gated receptors in the postsynaptic membrane. Now, the more neurotransmitter that is released, the greater the change in the potential of the postsynaptic cell. And the amount of neurotransmitter released is dependent on several factors, which we'll look at in the next few slides. But essentially, it is a combination of how many presynaptic neurons are signaling the postsynaptic membrane at one time. That's called spatial submation. In addition to how rapidly the presynaptic neuron is generating action potentials and releasing neurotransmitter. So if it's firing more rapidly, you're going to have a lot more neurotransmitter in the cleft as opposed to neurons that are firing occasionally. That is called temporal summation. Now the synaptic delay is about 0.5 milliseconds, which means that the more synapses there are in a neural pathway, the slower the rate at which the effector in the end of the pathway will be signaled. Also note that chemical synapses involve one-way information transfer from the presynaptic to the postsynaptic cell. Let's look at these postsynaptic potentials we mentioned on the previous slide. So when a neuron releases its neurotransmitter, it is called the presynaptic neuron. And the effects of that neurotransmitter can be either excitatory or inhibitory on the cell that is being signaled, which is called the postsynaptic cell. Excitatory postsynaptic potentials depolarize the membrane. So they're abbreviated EPSP for excitatory postsynaptic potential. It often results from the opening of ligand-gated channels under the influence of neurotransmitter binding. The influx of sodium makes the membrane less negative, more positive, thereby depolarizing it and moving it toward threshold. So excitatory postsynaptic potentials move the membrane in the postsynaptic cell towards threshold, and therefore the cell is more likely to experience an action potential. That is why it is excitatory. On the other hand, inhibitory postsynaptic potentials, abbreviated IPSPs, result from the opening of ligand-gated chloride or potassium channels. Now recall that chloride ion has a higher concentration outside the cell. So when channels for chloride ion open, negative chloride is going to flow into the cell and make the membrane even more negative, moving it away from threshold. Also, opening of potassium channels will allow potassium to flow out. The cell will be losing positive charge and will also be hyperpolarizing. So as the membrane potential moves away from threshold, these cells are less likely to experience an action potential, so the neurotransmitter effect in that case would be inhibitory. Now, as long as a neurotransmitter is in the cleft, it has the potential to bind to its receptor on the postsynaptic membrane and will lead to the postsynaptic effect. To control that effect, the neurotransmitter is removed from the synaptic cleft. Some of it moves out passively by diffusion, so it just drifts away out of the cleft and no longer is affecting the postsynaptic membrane as the neurotransmitter moves down its concentration gradient. Often, there is an enzyme that breaks down or degrades the neurotransmitter. So we have synaptic enzymatic degradation. We saw this with acetylcholinesterase that broke down acetylcholine at the neuromuscular junction. Also, many neurons have pumps in their synaptic end bulbs that actually take up the neurotransmitter that is released. It's called neurotransmitter reuptake. And again, it helps to regulate the amounts and duration of the effect of the neurotransmitter in the cleft. Note that glial cells, especially the astrocytes, also have the ability to take up neurotransmitter out of the cleft. That requires ATP. So an application here is a drug called Prozac. It is called a selective serotonin reuptake inhibitor. This drug prevents neurons from reuptaking the same amounts of serotonin that they have released in an untreated client. Serotonin is a hormone implicated in mood and a sense of well-being. So certain individuals diagnosed with depression may have low serotonin levels. One treatment is this drug Prozac. It blocks the reuptake of the serotonin so that serotonin stays in the synaptic cleft longer and binds to its receptors longer, and therefore creates a better mood for these clients. So all the effects have to be added up. We mentioned that the effect on the postsynaptic membrane not only depends on the type of neurotransmitter, but the effect of that neurotransmitter. Is it excitatory or inhibitory? Also, we have summation, the combined effects of neurotransmitter release either from multiple neurons or over a period of time. So summation might be spatial. With spatial summation, more than one presynaptic neuron is signaling a single postsynaptic neuron. Now, this is highly simplified. Of course, in your nervous system, we have multiple cells talking to each other at any given time, but this is just an illustration. Now, this is summation of the effects of neurotransmitters released from several end bulbs and several axons onto the postsynaptic membrane of one neuron. So if you add up all of that neuron activity within a given period of time, you get spatial summation. And if it's an excitatory stimulus, then that will definitely bring the membrane to threshold, more likely to get an action potential as multiple neurons are signaling at the same time, as opposed to individual neurons as shown in the first half of the graph. With temporal summation, we are looking at the effects of a single presynaptic neuron or end bulb releasing its neurotransmitter more rapidly or in rapid succession. And the buildup or accumulation of neurotransmitter is going to open more channels, and the influx of sodium in the case of an excitatory postsynaptic potential is going to eventually bring the membrane to threshold. Now, if that presynaptic neuron is releasing the neurotransmitter at a slow rate, then as the neurotransmitter is removed from the cleft, the effects are not going to bring the membrane to threshold. But if they release the neurotransmitter in rapid succession, it accumulates before it can be removed from the cleft and brings the membrane to threshold, leading to an action potential. So that's summation over time compared to summation in space with multiple neurons signaling a single postsynaptic neuron at any given time. We are wrapping up. There are different neurotransmitters released from the neurons of your body. Each neuron releases its own neurotransmitter, but note that a given neurotransmitter can be excitatory in one part of the body and inhibitory in another, depending on the type of channels they open in each area of the body. So neurotransmitters can be excitatory or inhibitory, and they are present in the axons of the central nervous system and peripheral nervous system. Important neurotransmitters include the following. All I want you to do is recognize the names. Acetylcholine, we saw that at the neuromuscular junction. Glutamate, aspartate, gamma aminobutyric acid, which is abbreviated GABA, GABA. We also have glycine, norepinephrine, and epinephrine are neurotransmitters, but they're also hormones released from the adrenal medulla. And you probably have heard of dopamine. You can add serotonin to the list as well. Now, research is still out on this, but it is known so far that axons and dendrites may be repaired if they're damaged if the cell body remains intact. If the cell body is damaged, that neuron is done. And remember, they're non-regenerative. So healthy neurons cannot divide to replace the ones that are damaged. However, they can be repaired if these conditions are met. So number one, the cell body remains intact. Schwann cells remain active and form a regeneration tube. Scar tissue does not form too rapidly to block repair and regeneration of the axon. And if the conditions are right, within several months, regeneration or growth of the axon can occur. But note that this is only in the peripheral nervous system. Axonal buds grow down the tube to reconnect, but grow at the incredibly slow rate of 1.5 millimeters per day. Now imagine that rate of regrowth when the axons that signal your feet and toe activity have their cell bodies up in the sacral and coccygeal regions of your spinal cord. So some of these neurons are more than a meter long. And if it's going to regrow at a rate of 1.5 millimeters per day, wow, that's slow. So take care of your neurons. Once they're damaged, they're hard to regenerate and cannot be replaced. In the central nervous system, formation of new neurons from stem cells was not thought to occur, but recent research suggests that distinct clusters in the brain, for example, the hippocampus, which is associated with memory, may undergo neurogenesis and regrowth. But there's a lack of neurogenesis in other regions of this brain and spinal cord. Neurogenesis is not as likely in the CNS as it is in the peripheral nervous system, as it is inhibited by neuroglial cells. There are low levels of growth stimulating factors in the central nervous system. The neurons lack neurolemmas, and scar tissue formation is quite rapid in the central nervous system. So if you're interested in regeneration of CNS tissue, I provided a reference at the bottom of the slide.